Thank you once again for inviting me. And my strategies I'm going to talk about today are drug strategies, but certainly forest bathing and all of that sounds very good for me if I had time because of the overwork problem. So these are my disclosures. So most of our drug targets are proteins, HMG co-reductase for statins, beta receptors, etc. But sometimes we can also target the protein directly, either decreasing its production or binding the circulating protein. And why would we want to do that? Although we need quite complex technology, it's often a little bit easier than looking for a small molecule. Because if you know the genetic sequence, you can edit, replace the gene, you can prevent the translation. If you know the protein, you can raise an antibody against it. While for small molecules, you end up screening large compound libraries, trying to find a drug that will work. So some of the novel strategies that we're going to be looking at today are either binding the protein with monoclonal antibodies or antibody-like proteins like the adnectins, newest PCSK9 inhibitor on the block, lerodalcibep, or we're going to be looking at RNA-directed therapy with antisense oligonucleotides or small inhibitory RNAs, or going for the DNA directly, either with gene or base editing or gene transfer. And just to look at some of these kind of technologies, I've said here fully human monoclonal antibodies. There's quite a couple of ways to get these antibodies using either transgenic mice, hybridomas, etc. It is important to get a fully human monoclonal antibody, as Pfizer learned with the experience with bococizumab, a PCSK9 antibody. And what you can see essentially here, patients developed anti-drug antibodies and lost efficacy. So you really do need an antibody that will not generate an antibody response to itself. Or antisense oligonucleotides. They are short nucleotides that very specifically target the messenger RNA of your target protein. So this antisense oligonucleotide will bind to the messenger RNA, and that whole complex will be degraded, and you'll be making less of the target protein. Or you can use a small inhibitory RNA, very similar concept. Again, you end up with translating less of that target protein. And one of the sort of real breakthroughs in this technology has been using hepatocyte targeting with GalNAc. GalNAc binds to the hepatic acylglycoprotein receptor very specifically, and with that you can ensure that your drug really gets delivered to the hepatocyte very, very specifically. And you can conjugate small inhibitory RNAs or ASOTs to this. And just to show you how that would work here for a small inhibitory RNA, GalNAc conjugated binds to that hepatic receptor, gets internalized. And to show you a real-world example, ASO volanesorsen and olezarsen, they're essentially the same antisense oligonucleotide. The one is GalNAc conjugated and the other one is not. And the dose you need is about 13% of the previous dose, with less side effects, obviously, if you use this type of drug. So gene strategies, either gene transfer, you see the evolution of adeno-associated viruses for gene transfer, or gene or base editing, and there's a whole lot of new technologies, CRISPR-Cas9 type technologies for this type of treatment. Now what I want to do is I want to take these technologies into some of the diseases that we see in the lipid clinic all the time: familial hypercholesterolemia, familial chylomicronemia syndrome, and high lipoprotein a, and just show you some selected applications of these types of technologies. So We've heard about familial hypercholesterolemia already today. These are some of the target proteins we might be wanting to attack. And first, the LDL receptor. Well, that was in fact one of the first publications of gene therapy in humans, trying to use ex vivo gene therapy with a retrovirus in this case, and essentially taking part of the liver, transfecting it with an LDL receptor, and then reimplanting those hepatocytes. And it was modestly effective, but not taken any further. There has been some studies with other viruses, unfortunately not that effective in this specific scenario. You all know about PCSK9 technology, and that has been extremely successful. So here we have PCSK9 inhibitors in heterozygous FH. You see the 50 to 60% reduction in LDL cholesterol. Homozygous FH, much less effective because patients often have very little residual LDL receptor activity. But there are some patients with homozygous FH that get clinically worthwhile responses to these PCSK9 inhibitors. So that's why the guidelines recommend you should always give the patient a therapeutic trial of this kind of drug. And then instead of binding the PCSK9 protein, we can of course prevent its synthesis and reduce the concentrations that way. And that is using a small inhibitory RNA. Inclisiran has actually made it to the market. So what you can see here is quite marked reductions in PCSK9 and then LDL reductions in the about 50% range with this drug. And its claim to fame obviously is for most of these small inhibitory RNAs, you have very infrequent dosing. So for this drug, you would give a dose at initiation, then 1, and then 3, and then 6 months later. So once every 6 months in the long term. Angiopoietin, protein like 3, ANGPTL3, being discovered as a cause of low cholesterol in an Italian small town. So if you've got mutations in ANGPTL3, you have what's called familial combined hypolipidemia, low triglycerides, low LDL, also slightly low HDL, and clearly very exciting drug target, and it's been shown to be functioning independently of LDL receptor activity. So we have evinacumab as a monoclonal antibody, and what you can see here, essentially 45 to 50% reduction in LDL cholesterol in patients with homozygous FH, and no difference according to the residual LDL receptor functionality. So a major breakthrough in the treatment of patients with homozygous FH. And it works in other patients with refractory hypercholesterolemia as well, although the approval is for homozygous FH only. And there's other ways to inhibit ANGPTL3. There's a small inhibitory RNA called zodasiran. And we have some early results from a proof of concept study showing about a 48, 49% reduction in LDL cholesterol with this drug. Unfortunately, the manufacturers decided to cancel its larger homozygous FH trial, and they are focusing their attentions elsewhere, which is very disappointing. And then, of course, the newest kit on the block, base editing. Let me just show you a proof of concept study. This is from mice. And essentially, the first thing you do is you inactivate the LDL receptor in mice. So you make them homozygous familial hypercholesterolemia, and you can see the LDL cholesterol goes up dramatically. And then you use a base editor to inactivate ANGPTL3, and once again, you now have a reduction in the LDL cholesterol in these mice. And the phase 1 trial of this drug is going to be starting soon. And the concept here is obviously what's called a once and done therapy. You would give these patients this therapy once using lipid nanoparticles, with, again, GalNAc conjugation, and then treat them successfully, hopefully. And we're all looking forward to seeing further results from this. So when we leave familial hypercholesterolemia, to briefly just look at familial chylomicronemia syndrome, severe hypertriglyceridemia, recurrent acute pancreatitis. Again, one of the first approved commercial gene therapies, alipogen teperovec, and the company, in fact, sold one commercial dose. That's all that they actually sold commercially. And you saw a temporary reduction in the triglycerides. The effect wasn't sustained, although there seemed to be less pancreatitis in the long term, and this drug is no longer available. You know, you had to give patients several hundred intramuscular injections to deliver the drug to the site of action, but certainly exciting and maybe something new will come. And then we've learned that reducing the concentration of ApoC3, although you would think it doesn't work in a patient who doesn't have functional lipoprotein lipase activity, it does seem to reduce triglycerides. And here we have initial data from the first volanesorsen, and you can see the reduction in triglycerides in the 40 to 50% range. And then the newer development, the olezarsen, again, quite marked reduction in triglycerides with this drug. And the big advantage of olezarsen is you're using a much smaller dose, so the drug is well tolerated, you don't have a lot of injection site reactions, and you don't seem to see the thrombocytopenia that complicated treatment with volanesorsen, especially in the patients with FCS. So certainly progress on that front as well. And then to look at elevated lipoprotein a, which has been a little bit of a stepchild in the lipid world, neglected for many years, but we are starting to pay attention to the stepchild and realizing the relevance and importance, especially of marked elevations in lipoprotein a, but we haven't really been able to treat it effectively except for lipoprotein a apheresis, which was really not done in many countries. But now we can target apoprotein a, and there's two ways to do it. One is with antisense oligonucleotides that decrease the production of apo and there we are going to have some results from a cardiovascular outcome study quite soon next year, hopefully. But just to show you the quite marked reductions you can get in lipoprotein a, and you can do the same with small inhibitory RNAs, and I'm just showing you the data for one of them, olpasiran. Here again, you have your much less frequent dosing, but there is others on the market, not on the market, in the development pipeline. So we've got several drugs targeting lipoprotein a that are coming up. And I want to end by saying that these novel strategies allow us to target previously undruggable targets highly specifically, and most of these drugs have a good safety profile. The delivery and the targeting systems are advancing, but we have the universal problem that cost of these interventions and drugs still precludes access for the majority of patients in need. And just to end off by saying, from IAS, EAS, we've been running the Homozygous International Clinical Collaborators Registry of Homozygous FH, and just polling the members there is quite frightening to see how restricted access to some of these novel interventions is in many, many countries. And that will really be a thing that I, and I'm sure Borger going forward, want to tackle, is that we have these excellent drugs and we need to get them to the patients. And thank you very much.